All right, hello everyone. This week we are going to analyze the data that we collected from last week. So, if you remember the scientific process, we start off with the observation, then we ask a question. From there, that question can lead us to a hypothesis. After we have formulated our hypothesis and prediction, we design an experiment, collect data, and last week, since we collected the data, in this week, we're going to analyze the data and draw a conclusion from our statistical analysis. All right, so this is right here. This is just hypothetical data. When you take a look at the Excel that your lab instructor has sent out to you, it'll be something very similar to this. So essentially, we are analyzing the data that we collected last week. Now, again, I would like to go over the mean, standard deviation, and the standard error of mean one more time. So the mean is the average, and the standard deviation measures how far each of the data points are compared to the average. So you take a look at these two examples right here. The average is shown in orange, and then the blue are all the data points. In this data set right here, the standard deviation is pretty spread out. While in this example right here, the data points are close to the average. So in this data set, since each data points are spread out, this data set would have a larger standard deviation compared to this one. Now, standard error of mean measures how far your sample mean deviates from the actual population mean. So as a reminder, the standard deviation measures how far your sample points are compared to the average. The standard error of mean measures how far the sample mean is compared to the actual population mean. So what does this two thing represent? So let's say, remember, we went out into the field and we collected data. We took the average of each of our sample. That is our sample mean. Let's say we were to repeat this experiment again. We would get another average. Now let's say we would repeat this experiment again, get another average, and then repeat the experiment again, get another average. The average of all these means right here, that is going to be your actual population mean. So the standard error of mean measures how far the sample mean, which is the one you get from each time you do the data collection, it measures how far the sample mean are compared to the actual population mean. So that is what was calculated in Excel. In this week's lab, we're going to focus primarily on biostats. We are not going into the theory of how these statistical tests are derived, but we're going to go through the basic steps of how to perform the statistical analysis tests and how to analyze your data. The reason why biostats or statistics overall is a very important field is because without math, if you take a look at certain data and you come to a conclusion without having math as your backup, you are essentially making a judgment call. So, for an example, if you take a look at these two plants, one plant was given fertilizer A, the other plant was given fertilizer B. In this example, at this point, you can just see that there's only one sample size. So, it's not a good experiment to begin with. But if you just draw the conclusion that fertilizer A is better than fertilizer B just because the plant here appears to be a little bit taller, you would be making a huge judgment call, and this is bias. So again, in order for this to be correct, you would obviously need more sample size, and you actually need to actually measure the length of each plant and then perform statistical analysis before you can derive to a conclusion. Now, if you were to come up with a conclusion, if in fact fertilizer A did produce taller plants than fertilizer B, if fertilizer A did indeed produce taller plants than fertilizer B, this is considered to be statistically significant. So your data are significant if after you perform the statistical analysis test and you get a significant p-value, that tells you that the difference that you see between the two treatment groups is really due to something in the fertilizer and that it's not happening by random chance. That is what statistically significance mean. So, first of all, before you start running any stats, it's good to take a few steps back and let's come up with our hypotheses for this test. So, let's say you hear around that Home Depot have a brand new fertilizer and it produces, really good plants, healthier plants. Okay? Your alternative hypothesis that plants that are given that new fertilizer, let's label it A, will be taller compared to plants that are given fertilizer B, which is what you typically use. This is the alternative hypothesis. The null hypothesis is the hypothesis that tests for no difference. So in this example, the null hypothesis would be that there's no difference in plant height between the two fertilizer. So make sure you understand the difference between the null and the alternative hypothesis. So from there, okay, you would design an experiment, carry it out, collect your data, and then you can use the correct statistical test to analyze your data. After you perform the statistical test, you're going to get a p-value. The p-value comes from you performing the test. The p-value is the probability of error value. So it's essentially a percentage. Since it's a probability, it's going to range from 0 to 1. The significant level, also known as the alpha level, is set to 0.05. So if you get a p-value that is less than or equal to 0.05, this means that your data are significant and that any difference that you see between the two groups are actually due to something contributing to that difference and it's not due to random chance. So if your p-value is greater than 0.05, this means that there is weak evidence against the null hypothesis. So if that's the case, your data are not significant. So you can pretty much accept your null hypothesis, and this also translates to fail to reject your null hypothesis. However, if the p-value is less than or equal to 0.05, this is strong evidence against the null hypothesis. So what you would do is reject the null hypothesis, accept the alternative hypothesis because your data are significant. If your p-value is less than or equal to 0.05, again, this means that there is something in your treatment group that is contributing to the difference. And at this point, you should see that the p-value is measuring evidence against the null hypothesis. All right, so let's take a look at the example that we did in class. So one of the examples that we did was measure light intensity between the shade versus the sun. Okay? As of right now, taking a look at this, you should know that the hypothesis is incomplete because I didn't provide a rationale. So remember, in your hypothesis, make sure you have a good explanation why. So let's take a look at our alternative hypothesis first. Okay? The alternative hypothesis is the hypothesis that tests for a difference. So we hypothesize that light intensity in the sun is going to be greater than light intensity in the shade. This is one way that you can write it. The null hypothesis is the hypothesis that tests for no difference. So here, light intensity in the sun is going to not be equal to light intensity in the shade, or you can write it this way: the null hypothesis is the hypothesis that tests for no difference. So the null hypothesis here would be light intensity in the sun is equal to light intensity in the shade, or you can actually write it the opposite as the alternative and say light intensity in the sun is less than or equal to light intensity in the shade, but there has to be an equal component to it because remember, the null hypothesis is the one that tests for no difference. So let's say you put this into Excel and you're going to get a p-value. Okay. Now the p-value that you're going to see from Excel, you're actually going to get two p-value. One p-value says one tail, the other p-value says two tail. you need to know the difference between the one tail and the two tail p-value because you have to accept the correct one. Okay, so although you're getting two p-values, you have to choose the right one to analyze your data, to base your conclusion from. The one tail p-value is when you have a hypothesis that is directional. In this case, take a look at your alternative hypothesis. You hypothesize that light intensity in the sun is greater than light intensity in the shade. So there is some sort of directionality to here. If that's the case, you would take a look at the p-value for one tail. However, there are many studies where people are just trying to see whether there's a difference or not. Let's say here you hypothesize that light intensity in the sun is not equal to light intensity in the shade. You did not give any direction. If that's the case, you would take a look at the p-value for the two-tail, the non-directional one. So whichever way you worded your hypothesis, specifically the alternative hypothesis, choose the p-value that correspond to it. There are many research where scientists are just testing to see whether there's a difference or not. This can be clinical research where they are coming out with a novel drug. They don't know if that drug is going to have any effect. So they just want to see if the drug has an effect or not. That would be a two-tail experiment. But in our case, we are most likely going to take a look at the p-value for the one tail because there is some sort of a directionality in the way you word your hypothesis. So let's say you perform the analysis test and you get a p-value that is 0.2. 0.2 is greater than 0.05, which is the significant level. If that's the case, what you have to do is you are going to accept the null hypothesis because your data are not significant since this p-value is greater than 0.05. However, if your p-value is 0.03, this is below 0.05. This tells you that your data are significant, that whatever changes that you see are actually due to something in the treatment group and is not due to random chance. If this is the case, if your p-value is below 0.05 and your data are significant, then you can go ahead and accept the alternative hypothesis. If your p-value is greater than 0.05, your data are not significant and you would accept the null hypothesis. All right, so we talked about what the p-value means and how to analyze it. Let's again take a few steps back. There are many statistical tests. Okay? You have to choose the right statistical test to perform based on your data. So you can either have discrete data or continuous data. Discrete data is data where you are counting something and you can categorize these things. So if you're counting, if you're getting whole number and you want to take a look at different categories, so if you want to see how many birds prefer to live in tree A versus how many birds prefer to live in tree B, also how many female students are in STEM versus how many male students are in STEM. You're essentially counting, you have whole numbers and you're putting things into categories. That is discrete data. So if you have discrete data, you would be performing a chi-square test. If you have continuous data where you're measuring something, you would perform one of these tests. Now, let's say you're just looking at two groups of independent sample. Let's say you're just looking at light intensity in the sun versus light intensity in the shade and whether there's a difference between the two of them. Then you would perform the t-test. Now, let's say you have two or more groups of independent sample. Now, let's say you want to measure light intensity in the sun versus light intensity in the shade versus light intensity in the classroom. So here you have more than two groups of independent sample. You would do a one-way ANOVA. Now, let's say if you want to take a look at the relationship between two variables to see if something is correlated or to see if one variable affects the other, you would do either a correlation or a regression test. We'll talk about this in a bit. Now, the interesting thing is that if you were to perform the wrong statistical test, you may get the wrong p-value and your conclusion may be completely off. If you click on this link right here, it takes you to an article where scientists, they are working with the same data sets, but they perform different statistical analysis tests on the data set and they actually got different results. So you have to understand and know which statistical test to perform so that you don't make this, so that you do not make any mistake, and that the p-value that you get does reflect your data so that you can draw the appropriate conclusion. All right, let's go ahead and do this example as a class. Let's take a look at soil temperature between the sun versus the shade. So one of the abiotic factors that you measured last week was soil temperature. When you measure soil temperature, you are looking at the pH probe, and this is a type of continuous data. And specifically, if you're measuring soil temperature between the sun versus the shade, there are only two groups of independent samples here. So essentially, you would be performing the t-test. Now, before you can actually perform the t-test, you have to perform the f-test. The f-test needs to be conducted before the t-test is performed. Now, when you perform the f-test, you're going to get a p-value as well. But please understand that the p-value that you get from the f-test, it does not tell you anything about whether your data are significant or not. All that it tells you is whether your variances are equal or unequal. This tells you whether your data are not widely spread or your data are widely spread. So let's say you get a p-value from the f-test that's greater than 0.05. Essentially, your variances here are equal. Your data, they're not widely spread. So the data points are clustered close to each other. If that's the case, you're going to do a t-test assuming equal variances. However, if you perform the f-test and you get a p-value that is less than or equal to 0.05, this tells you that your data are widely spread, your variances are unequal, so you would do a t-test assuming unequal variances. So with that, you then know which t-test to perform. From the t-test, you get the p-value. Remember, you're going to get a p-value that is 1-tail and a p-value that's 2-tail. Make sure you choose the right p-value to look at depending on how you word your alternative hypothesis. But regardless, the p-value that you get from the t-test, that is going to be the one that determines whether your data are significant or not. The significant level is set at 0.05. So if your p-value is 0.05 or less than that, your data are significant. Whatever difference that you see between the groups are due to something that's in the group, not due to random chance alone. So if that's the case, you can go ahead and accept the alternative hypothesis. All right, so again, these are just hypothetical example. Let's take a few steps back. If you're taking a look at these data, okay, you are essentially measuring soil temperature. When you're measuring soil temperature, these are continuous data. Again, you have two groups of independent sample, so you're ultimately going to do the t-test. But before you do the t-test, you have to do the f-test. So the f-test has to be done first. Okay, the f-test will tell you a p-value. Remember, the p-value that you get from the f-test does not tell you anything about whether your data are significant or not. It does not tell you which hypothesis to accept. All that it tells you is whether your variances are it just tells you whether your variances are equal or not, and then which t-test to run. So from the f-test, it'll tell you which t-test to run. The t-test is what you run last, and from the t-test, you will get a p-value as well. Choose the right p-value, whether it's 1-tail or 2-tail. The p-value that you get from the t-test, that will tell you whether your data are significant or not. Okay. All right, so let's go ahead and let's take a look at a practice example together. All right, so this is a practice example. Okay, your data may be different from this. Okay. All right, so as a reminder, this is what your lab instructor showed you last week on the board, they should have calculated the mean, standard deviation, and standard error of mean. And when they email you this data, they should have set up the Excel for you. So let's go ahead and take a look at soil temperature, which is what we saw. So in the second tab right here, go ahead and click on soil temperature. Okay, so pretty much all that we've done is just copy and pasted the soil temperature data here over to the second tab. Okay. All right. So what we are having, what we see here, are going to be continuous data, and you have two groups of independent sample, so you're going to do a t-test. But remember, before you do the t-test, you have to do the f-test. So to perform the f-test, you're going to go to data, data analysis, click on f-test, click OK, and then the variable 1 range, you're going to click here and highlight just the numbers, just the number for soil temperature under the sun. Click back here, then click on this. For the variable 2 range, you're going to highlight the values, just the values under soil temperature in the shade. Okay. When you've done that, for the output range, this is where you want your data to be stored. Click on the output range, click back on here, and I like to store my data just here. So I just click on E1. I do not recommend that you store your data in a new worksheet. That's going to get messy. Okay, so just store your data in the same worksheet. Then click OK, and this is the result from the f-test. Now go ahead and take a look at the p-value. Remember, the p-value that you see here does not tell you anything about whether your data are significant or not. All that it tells you is whether your variances are equal or not. In this example, the p-value from the f-test is 0.09. 0.09, okay, is greater than 0.05. So your variances are equal. Okay, this means that your data are not widely spread. Okay, so let's go back here and take a look at the Excel. Okay, so see how here from the f-test it says that if the p-value is greater than 0.05, that means your variances are unequal, your data are not widely spread. So then you would do a t-test assuming equal variances. So let's go back to the Excel. Okay. All right, so since our p-value here is greater than 0.05, we're going to perform a t-test assuming equal variances. So you go back to the data tab, and then you click on data analysis again. But now you're going to finally do the t-test, and you're going to do the t-test assuming equal variances because the p-value here is greater than 0.05. Then click OK. For your variable 1 range, same thing as you did before, it's going to be the values under the soil temperature in the sun. For your variable 2 range, it's going to be the values under soil temperature in the shade. So remember, only highlight the values, the numerical value. For your output range, you're going to put it here, okay, or anywhere on this worksheet. I just recommend you do not put it in a brand new worksheet because then it'll get messy. Then click OK. Okay. Now, if you take a look for the output of the t-test, you see that there are two p-value. There's a p-value for one tail and a p-value for the two tail. Now, if we take a look at how we worded our alternative hypothesis here, most likely you hypothesize that soil temperature in the sun is going to be higher than soil temperature in the shade. Because there's a directionality to this, you said that it's going to be higher in the sun compared to the shade. You provided a direction. In that case, you're going to look at a p-value for one tail because you provided a direction. So this is the p-value that you're looking at here. Okay. All right. So with this p-value, you can see that it is 0.02. Since it's 0.02, or you can round it up 0.03, this number is less than 0.05, so your data are significant. This means that whatever difference that you see here, the difference between the soil temperature, it's actually due to something contributing to that difference. It could be the sun, okay? It's not due to random chance. So if that's the case, you can go ahead and accept your alternative hypothesis based on this data, based on your p-value. Soil temperature in the sun is higher than soil temperature in the shade. All right. And then if you take a look at the other tabs, okay, go ahead and perform the correct statistical analysis for the remaining part of your data. So you would need to perform the F-test and the T-test for soil temperature, air temperature, soil pH, soil moisture, and light intensity. Okay. Now, there may be one of these abiotic factors where you predict that there is no difference. Okay. If that's the case, if you just predict whether there's no difference or if there's difference, remember, instead of looking at the one tail, then you would look at the two tail if that's the case. Right. Now, I'm going to skip to the next step. Okay. This right here, okay, is a type of inferential data where you perform statistical analysis and you get a p-value and you come to a conclusion. But with statistics, you can also have descriptive statistics. And with descriptive statistic, you can actually graph your data. So we can actually make a bar graph for the temperature of the soil temperature in the sun versus the shade. So to make a bar graph, you are essentially just graphing the average. So go ahead and highlight these four cells, okay? Make sure you are including the sun and the shade title as well. And then all you need to do is then go to Insert, click on the graph here, click on 2D column, and then here you go. You have your bar graph. But of course, we are not done yet. We're still missing a lot of things, okay? We're still missing the error bars. So to do the error bar, you're going to click on one of the bar and then it'll highlight both. Then you go to Add Chart Elements, go down to Error Bar, and you want to customize your error bar. If you just click here, it'll give you a random generated one, which is incorrect. To customize it, go down to More Error Bars Options, and then you're going to click the Custom right here. Click on Specified Value. For the positive error value, you're going to highlight the standard deviation. We're just going to work with the standard deviation here. For the negative error value, you're again going to highlight the standard deviation. And when you do that, okay, you have your own customized error bar that is according to your data set. But again, we're still not done. Our bar graph is missing a title. Okay, so I like to base my title off of the p-value. So p-value here, since it's significant, we hypothesize that there are some significant difference between the soil temperature in the sun versus the shade. Okay, so an example of a hypothesis would be soil temperature in the sun is higher than the shade. Okay, or if that's too long, you can write significance different in soil temperature between the sun and the shade. Okay. So you can also write soil temperature in the sun versus soil temperature in the shade, which is what your lab manual provided. But in my opinion, a better title is the one that is straightforward and reflects the finding. Now, you can go ahead and include the sample size. Usually you would see the sample size in the figure caption, but because you're not writing a figure caption, go ahead and just put the sample size in your title. The sample size here, okay, since we have 9 samples under each treatment, the sample size would be 9. Okay, it's not 18. You don't add both of these together. It's how many sample you have under each treatment group. And we are still not done. You're missing the axis title. So to get the axis title, you go to add chart element, axis, primary horizontal, axis title, primary horizontal. Okay, sorry. So go to add chart element, axis title, primary horizontal. This right here, you can say it as treatment, you can say it as conditions or weather conditions, etc. That could be one possibility. And then you also need to do the axis title for the vertical. So the axis title for the vertical is again going to be soil temperature, and make sure you write the units, which is in Celsius. Okay. All right, and then if you want to get fancy, you can change the color for each bar graph so you have two different colors. Now, again, if you take a look at these two data sets, you may see that the numbers are pretty close. But when you actually run the statistical analysis test, it tells you that there is a difference. The p-value is much lower. So your data are significant. There is a difference. If you just take a look at the average, one is 20.28, one is 19.26. You're like, oh, they're pretty similar. But again, that is making a judgment call. So if you did not run the actual statistical analysis test, essentially you wouldn't know. Okay? You would probably conclude that there's no difference. But after performing the statistical analysis test, there is a difference. All right, so that is the example for soil temperature. You're responsible for doing air temperature, soil pH, soil moisture, and light intensity in the lab for this week. Let's go ahead and go back to the PowerPoint. Okay, and remember the directions for everything, it's in your lab manual. The directions for the bar graph too, you're still confused, it's also in your lab manual. Now let's move on to the next part of the lab. So far we talked about the t-test. Let's go ahead and transition to if you want to take a look at the relationships between two variables. Here you can do a correlation or a regression test. So for the correlation, you just want to see if two variables are correlated to one another. You're not looking to see whether one variable affects the other. An example of a correlation test is you want to see if the height of a person corresponds with their weight. So taller people tend to weigh more, but being tall does not cause you to be heavy. But essentially, taller people do weigh more. This is an example of a positive correlation. You can also take a look at a negative correlation as well. So if you take a look at the number of absences for our students and their grade, what you may see is that the more absence that student has, the lower their grades tends to be. And that is a negative correlation. But at the same time, remember, being absent does not cause you to have lower grades. Okay, it just somehow is correlated. Okay, there are other factors that are involved, obviously, but overall, for correlation, you're just taking a look at the relationship between two variables. Sometimes you can take a look at the relationship between two variables and there's no correlation. So you want to measure the amount of tea someone consumed versus their intelligence level. You may see that there's no correlation at all here. So this is a correlation test. Now, the regression test is when you want to see if one variable affects the other. So you're going to have an independent variable and a dependent variable. If you want to take a look at the effects of sunlight exposure on seed length, you want to see whether plants that are exposed to more sunlight, okay, will have longer seed length. So pretty much your independent variable in this case is sunlight. The seed length would be the dependent variable. If you want to see how sunlight affects the seed length, then you would perform a regression test. So let's say you were to actually run this test, okay? And let's say you got these data, okay? What's really cool is that you can actually take this data and make a line graph out of it, okay? Since the sun exposure, the amount of time it was exposed to the sun is the independent variable. It's going to be on the x-axis. The seed length, okay, since we hypothesize that it may be dependent on sunlight, that's going to be on the y-axis. So the independent variable is on the y-axis. You can also make a you can also make a trend line. You can also add a trend line to this, and the trend line will give you the equation of the line. So pretty much all the data points that you see, the trend line is the best fit line that tries to go through all the data points. Okay, and pretty much it's y equals mx plus b. Okay, so you got the slope right here and the y-intercept. You can also figure out the r-square value as well when you use Excel to make a trend line. Okay, so the cool thing about the equation of the line is that it tells you the slope and y-intercept. But in addition to that, let's say, right, if you take a look at your data set, let's say later on, okay, you find a seed that was 2.1 millimeter or millimeter, okay, and you forgot to write down the time that that seed was exposed to the sunlight for. So you have a seed length, but you forgot to record the data of how long that plant was exposed to the sun. Using your trend line equation, you can just plug in the length of that seed into this equation. So since that seed length is the dependent variable, you would plug it into y. Okay, so go ahead and you can plug it into y, and then to find out how long that plant was exposed to the sunlight for all you have to do would just solve for x. Okay, so if you do that, okay, after you solve for x, you should get an estimate that that plant was probably exposed to the sun for around 16 hours. So again, the trend line can allow you to plug in one variable and solve for the other. However, you are also going to get the r-square value. The r-square value ranges from 0 to 1. The r-square value tells you how close your data points are to the trend line. The closer your data points is to the trend line, it's going to be closer to 1. So that's the first thing that the r-square value tells you. If you get an r-square value of 0.8 or above, the data points that you see here are very close to the trend line. The second thing that the r-square value tells you is if you can use one value to predict the other. So in the previous example, we plugged in y and we solved for x. Okay? If your r-square value is high, you can do that, and whatever answer you get, it will most likely be reliable. However, if you have an r-square value that is very low, sure, you can plug in y and solve for x, but it wouldn't be as reliable. So for this example, our r-square value is 0.5. It's not close to 1. So the data points aren't really close to the trend line. Sure, you can plug in y, you can plug in the length of the seed and solve for x to see how long that plant may have been exposed to the sunlight for. But whatever value that you get, it probably won't be as reliable because the r-square value is so low. So that is what the r-square value tells you. It tells you these two things. Okay. All right, so now the second part of our statistical analysis test is to see the relationship between light intensity and each abiotic factor. The first one you're going to look at is the relationship between light intensity and soil temperature. So essentially the question here is, does light intensity affect soil temperature? Your alternative hypothesis now would be that light intensity does affect soil temperature. We hypothesize that as light intensity increases, soil temperature can also increase. And then remember, your null hypothesis is the one that tests for no difference. So here, light intensity does not affect soil temperature. You would perform a regression test. The reason why is because light intensity is the independent variable and soil temperature is the dependent variable. In this example, we want to see if light intensity causes soil temperature to change. We're not looking at whether there's a positive or negative correlation here. Here we have an independent variable being light intensity and a dependent variable being soil temperature, so we're going to perform a regression. You will also get a p-value from the regression test as well. If your p-value is greater than 0.05, this is weak evidence against the null hypothesis. So you can fail to reject the null hypothesis, which also means that you can accept the null hypothesis. Your data are not significant. Light intensity does not affect soil temperature. However, if your p-value is less than or equal to 0.05, this is strong evidence against your null hypothesis. You can go ahead and accept the alternative hypothesis. Your data are significant. Light intensity does affect soil temperature. So please make sure you understand what the p-value means. Remember, significant is set at 0.05. Anytime you get a p-value that is less than or equal to 0.05, your data are significant. You can go ahead and accept the alternative hypothesis. You will also get an R-square value from the regression test as well. Remember, R-square value tells you two things. R-square, first of all, ranges from 0 to 1. It tells you how close each data points are compared to the trend line. And the second thing it tells you is if you plug in X and solve for Y, or plug in Y and solve for X, how reliable would that be? The closer your R-square is to 1, the more reliable that would be. If your R-square value is very low, you can plug in X and solve for Y, you can plug in Y and solve for X, but the estimation wouldn't be as reliable. So remember, R-square value tells you two things. Okay. All right, so let's go ahead and do the relationship between light intensity and the soil temperature. So let's go back to Excel. Go ahead and click on the 7th tab. It is set up right here. Light intensity is here and soil temperature is here. Okay. So then you go to data, data analysis, and now you're going to perform a regression test. Click OK. And remember, the reason why it's a regression test is because you have an independent and the dependent variable. For here, make sure you input the correct value into each range. The input Y range, that is going to be your dependent variable. So that's going to be your soil temperature. The input X range, that's going to be your independent variable. So that's going to be your light intensity. So the Y range is your dependent variable, which is the soil temperature, and the X range is your independent variable, which is the light intensity. For the output range, again, I choose to put it in the same worksheet. I'm going to put it here. I highly recommend that you do that as well. Then click OK. So from here, it tells you the p-value. You're going to take a look at this p-value right here. You're going to take a look at this p-value. Okay. And right off the bat, you can see that this p-value is much greater than 0.05. So our data are not significant. You can conclude that light intensity does not affect soil temperature. It also tells you the slope, okay, and the intercept. So I'm going to highlight these two values here as well. Okay. Now, let's go ahead and graph this. Let's make this into a scatter plot, and you can also see the R-square value is here as well. So to make this into a graph, we're going to make a scatter plot. All you have to do is highlight these values. Okay? And if you set it up like this, if your lab instructor set it up like this for you guys, it's going to make it very simple. So to make a scatter plot, highlight the values, go to Insert, Chart, but now instead of doing a bar graph, we want to take a look at the relationship. So we're going to make a scatter plot. It's going to pop up something like this. Okay. Click on one point and it highlights everything. Okay. So click on one point, it should highlight everything. Okay. So then you are going to go to Add Chart Elements and then go to Trend Line and then go to Linear. Okay. There you go. You have a trend line. Then click on that trend line Sorry, give me 1 second. Add chart element, trend line, none, trend line. Linear. Okay. And then you're going to click on that trend line. Okay. And then right click. So you click on that trend line and then do a right click. Click Format Trend Line. And then you can choose the display equation on the chart, display R-square value on the chart. And then here you go. You see the equation of the line and the R-square value. Now this is very important because if you were to perform everything correctly, you would see that the slope matches to what you have here. Okay. And so does the intercept. Here it matches with what you have here. And the R-square value here matches to what you have here as well. So if you perform your regression test correctly and you did the scatter plot correctly, the trend line correctly, everything does match up. If something is off, you know that you probably switched the data range somehow when you input it into the regression test. Now, we're not done yet. We still need to to finalize this graph. Your chart title, this is just an example. I like to choose a title that reflects the finding. Since the p-value is high, it's not significant. So we can conclude that light intensity does not affect soil temperature. Okay? But please keep in mind, okay, and we'll talk about this in lecture 2, light intensity from what we know should affect soil temperature. The hotter the air is, the more heat radiation it gets. The soil temperature should be hotter. But in our example, in this specific example, we don't see that. And there are many reasons why. It could be, remember, we have a very small sample size. Maybe not everyone was measuring properly. Maybe the weather condition was off that day for some reason. So just because in this example you did not get significant data doesn't mean that this transferred to what is happening in the real world. It's just based on the data that you have here at this point in time. And then I'm also going to include the sample size. And in this example, the sample size is going to be 18, okay? Because there are 18 points here. We're almost done. Let's go ahead and finish up this graph. Let's go ahead and add our axis title. So I'm going to go to chart design, add chart element, axis title, primary horizontal here. You are looking at light intensity. And you want to include the unit, you can do the mu sign or you can write micromoles. It's in micromoles. And then for the y-axis, we are measuring soil temperature. Okay. And this is your completed scatter plot. Again, if you still need to review, okay, make sure you take a look at the directions in your lab manual. It tells you how to make the scatter plot as well. In lab, you're going to finish up the rest to take a look at light intensity, to see if it affects air temperature, to see if light intensity affects soil pH, and if light intensity affects soil moisture. And for all of these, you're going to do the regression tests. So you can complete the statistical analysis in class. As a reminder, for this week, you do need to bring a scientific calculator to class. It has to be scientific, and please bring your laptop with the Excel installed as a desktop version, not the browser. It's not going to work, and you should have your data analysis tool pack installed as well. If you're not bringing your laptop, then save the data that your lab instructor emailed to you on a flash drive, and you can use the school's laptop that would be provided for you in lab. And then here are the study questions for your ecology exam. Okay. All right, and that is it for this.